Hello, everyone, and welcome to another exciting ICPS webinar. My name is Kenny Coogan, the Education Director, and we're excited to have Leonora here talking about things living in pitcher plants. But first, I wanted to tell you a little bit about the ICPS, which is the International Carnivorous Plant Society. We have been around for over 50 years, and our growing membership includes professionals like Leonora, researchers, but it also includes hobbyists and people who are working in carnivorous plant nurseries. We are truly an international society, and we are really promoting carnivorous plant conservation these days, not just the cultivation. We like to have a webinar every month, but sometimes we're lucky enough to have more than one a month. In January, we already have two presentations for January and February in 2025, but we're going to have a full lineup for 2025 coming out very soon. And if these webinars are not enough for you, the first Wednesday of May every year is World Carnivorous Plant Day. And Wednesday, 7th of May, 2025, it's the 5th anniversary of World Carnivorous Plant Day, which means we have 4 years of World Carnivorous Plant Day for you to go back on and look at and study. You can find over 24 videos for each year on our YouTube channel, as well as our Facebook. And then we have the links on our Instagram. But if you go to our YouTube channel, you can see probably up to 100 videos now from scientists, from researchers, from people doing field trips just for fun, for people growing carnivorous plants on their windowsill. And we have this huge library of content that is all available for free. Like I mentioned, the first Wednesday of May is always World Carnivorous Plant Day. And if you would like to participate for the 5th anniversary, we are looking for content that has to do with the number 5. So we're looking for people just like you who are watching this webinar to give us a video that says 5 easy plants to grow indoors, or 5 fertilizers to use on carnivorous plants, or 5 fertilizers to use on Nepenthes, or 5 pet store foods that you can use on carnivorous plants. So we're looking for content that has the number 5 in it. If you're interested in presenting for World Carnivorous Plant Day, or if you're interested in presenting one of our webinars, you can send me an email at kenny@carnivorousplants.org. And with that, Leonora, thank you so much for presenting for our International Society, and you can share your screen. And also, thank you everyone attending this live. If you are attending this live, we highly encourage you to use the chat function at the bottom of the screen to ask Leonora very difficult questions, and I will ask them to her at the end of her presentation. So thank you, Leonora, and you're good to go.
All right, thank you. Thank you so much for inviting me to give a talk, and thank you everyone for being here. I'm excited to talk to you a bit about some of the research that I've been doing over the past 10 plus years into microbial communities living in carnivorous pitcher plants. I'm currently an assistant professor at Boise State University in Idaho. So I just wanted to start out by talking a little bit about the importance of plant-associated microbes, because it's really this broad field that we are still learning so much about. And I think many of you realize that fungi and bacteria can be plant diseases. They can attack plants and have these really negative effects. But we also have so many microbes that can have these positive effects on plants. And this slide is from a review that's just showing a bit about all the different things that are affecting plants that live, or microbes that live in association with plants. And so they're influenced by the environment, by species interactions, by plant host characteristics. As I was saying, they can have negative or positive effects on the host. So we know that there's a lot of plant pathogens, but we also know that there's things like mycorrhizae that some plants can't grow without. And they can affect plant growth, survival, and their stress tolerance in different environments. We're starting to learn more about how Leaf-associated microbes might help with surviving drought or osmotic stresses, like if they're in salty environments. And so there's a lot of different roles that these organisms play, and we're really still in the infancy of learning all about the different things that they do and the different ways that they interact with each other and with their hosts. And one of the things that my lab group is really interested in is broadly thinking about what shapes microbial communities and the ways that they function. And so we know that microbes are these tiny organisms, but have these huge effects on our planet. They can affect the biogeochemical cycles. And we still don't know a lot about sort of why different microbes might be found in certain environments, why you might have communities with so many different species that can all live together, and what are the most important factors that really shape what these communities look like. And so a lot of the processes that we know are acting on these communities are things like dispersal. You start with a regional species pool, and then a subset of those organisms will be able to disperse into a new environment. Then only a subset of those will be able to survive and thrive in a particular habitat, and that's environmental selection. And then you can also have these chance events that can be things like a big snowstorm or other kinds of weather events, or even just like which microbe happens to arrive first can change the composition of a particular community. You can have processes like succession changing over time, maybe through species interactions where you have some organisms that are good kind of early. They get to places early and they're good to establish in a new habitat, but then those might give way as better competitors are better able to survive and thrive in that community. And then over longer time scales, we have evolution acting and changes in different strains that can change their functional abilities, that can change the composition of communities. So all these different things are affecting what communities we see where and why, and also affecting the functioning, so the ways that they're able to act within different ecosystems. And one of the main things that we study in my lab group is carnivorous pitcher plants. And many of you, I think, know a lot about pitcher plants, but for those of you who might not know as much, they're modified leaves. So over kind of long evolutionary time, they've modified portions of their leaves to have this cup-shaped vessel, and they produce nectar around the lip, and sometimes under this lid, and that attracts insects, often ants, that come and feed on the nectar, but then they slip and fall and drown in the pool that's inside these little pitchers. And that pool of liquid is mostly rainwater, but can also have some plant secretions in there. And inside that pool, we find these whole little ecosystems. So you see that there's aquatic insects that are only found in association with pitchers, but there's also things like protozoa and rotifers and fungal yeasts and bacteria that all live together in these little pools. You have predators and prey, you have primary producers and decomposers. So you have all these different components of an ecosystem, but on a nice small scale. The other thing that's cool about pitcher plants is that they're generally sterile or very close to sterile before they open. So this lid kind of opens and then it's passive. It's not like a Venus flytrap where it moves around, but it opens, and once that pitcher opens, it then starts acquiring a community from its environment. And we think that a lot of the microbes, that microbiome that forms in there, is really helping the plant to break down and digest its prey. And so in a lot of ways, it's similar to a gut, right? We also are born close to sterile, but then very quickly as we're being born and very soon after, acquire some microbes from our parents, from those surrounding us, from our environment. And those microbes that colonize our gut help us to break down and digest our food. And so there's a lot of nice parallels here when you think about pitcher plant and a gut. Pitcher plants, for many of these reasons I've been talking about, are this emerging model system for community ecology, for being able to understand those different processes I outlined of all the different things that affect how a community forms. And so I'm just going to go through. They have these modified leaves that last up to 2 years in some species. And so you can actually follow them over time, which is nice. They capture insect prey, probably mostly for nitrogen, but also phosphorus and other micronutrients that they might need to grow. They contain this whole little ecosystem inside of them, which is very cool. They have a nice size and they have a clearly defined community. So they're like these little aquatic islands. And, you know, we may want to study all the microbes that are in like the whole rainforest, but you can imagine that the millions of species that might be there is not very tractable for being able to really understand what's going on in an ecosystem. So it's nice to have this smaller, clearly defined community. They're sterile before they open, or at least close to sterile, depending on the species. And they're easy to manipulate. You could imagine if you wanted to study something like, you know, a mouse gut, it's pretty hard to like add in different microbes and like sample from it over time. You usually have to sample the feces, right? Which is not necessarily what's going on in the gut. And so this is actually a much easier system to be able to study. And they're naturally replicated. So that means that, you know, one particular plant usually has many pictures on it. And there's many plants within a certain bog or habitat. And so you have lots of different pictures that are all growing together in the same ecosystem. And you can sample from those and get a sense of how they differ from each other, even within one site. As I was mentioning, they have this host microbiome association. And because of that, they also have this clear ecosystem function where we think that, you know, at least from the microbial component, it's probably really important for them to help degrade insect prey, right? That is sometimes in ecosystems, it's difficult to say, like, what are the important roles in this ecosystem that we want to study and that we know are really relevant for the health of the whole ecosystem. And in this little microcosm, right, we know that that breaking down that insect prey is where a lot of the nutrients for the whole community are going to come from. And so that's something that is really relevant and important for these little ecosystems. This is a little bit of an older plot, but I just like it because it's got all the pictures of the plants. But we know that carnivory has evolved in plants many different times, right? And usually it evolves in places where there's a lot of sunlight and a lot of water, but there's not a lot of nutrients in the soils. And so instead of getting their nitrogen and phosphorus and other micronutrients just from their soils and through their roots, these plants are getting those nutrients from capturing and digesting insects. And I want to highlight the pitcher plants on here. So we have the Nepenthes. These are in Southeast Asia. There's now over 100 species that have been described. And then there's the Cephalotus. This is a group that's found only in Western Australia, and it's not even really a group because there's actually just one species in the whole family. And then there's the Sarracenia, and these have 3 different genera. They're found in the Americas, from South America up into Canada, and they have a few different species in each genus, except for just one in Darlingtonia. And so I want to highlight that these each are in different orders of plants. So they're really distantly related. This is a clear example of convergent evolution, where you have the same form and function evolving totally independently in different lineages. And I'm just highlighting these. There's other things we might also call pitcher plants, but I highlight these as the sort of like true pitcher plants, where you have one pitcher formed by a single leaf, because that it is more like the same form. And they all have this function of trapping and digesting insects, usually as pitfall traps, but not always. And so this is this really nice example of convergent evolution, where you have many different, separate, independent lineages that have evolved very similar form and function. So for this talk today, I'm going to have 2 different parts. The first part is going to be, I think, a little longer, and it's talking about convergent interactions and host specificity between pitcher plants and their associated taxa. And then the second part will be more about understanding microbial functioning in pitcher plant microcosms. So thinking about this first part, when I was doing my PhD, I wanted to just understand more about like what organisms live in these pitchers. And at that time, there hadn't been much about the microbes in particular, because you need to really study those through nucleic acids, through DNA analyses. So we knew a little bit about the different insects that you might find living in pitchers, but not a lot else. And so we collected over 700 different samples in North America, Southeast Asia, and Australia from pitchers, and also a few from their surroundings. So from the bog water or the soils that they were growing in, to be able to also understand the habitats and the microbes that were in those habitats. Then we did metabarcoding of the DNA of both the bacteria using the 16S barcode marker and eukaryotes using the 18S ribosomal RNA barcode. And then we also did some shotgun metagenomes from a few samples. And that's where you basically, break up the DNA from all the different genomes from all the different organisms that are growing together in one pitcher. And then you can sequence chunks of those and you get a sense of the different functional genes. So not just who's there, but a little bit more about what they might be doing, what their capabilities are in their genomes. So this is a picture. One of the places I got to sample was Maleo Basin in Borneo. And it was this amazing opportunity to be able to go and see these gorgeous Southeast Asian rainforests. We have to wear leech socks when you're sampling there because they have these ground leeches that crawl up your legs, which is a little scary. But other than that, it was so fantastic to be able to be in this ecosystem and to work with some Malaysian graduate students as well. So our results from these kind of big survey is that, in terms of the eukaryotes, a lot of this, we knew a bit about the insects already, but there's mainly mosquitoes, midges, also mites, fungi, protozoa, and algae that are all growing in these pitchers. And in terms of the bacteria, it was mostly proteobacteria, Bacteroidetes, actinobacteria, and acidobacteria. So we got a little bit more of a sense of what are the different things that grow in these pitchers and to what extent do we see them showing up regularly. I'm going to talk first about this main comparison we did, which was between Nepenthes and Sarracenia. And just to highlight how distantly related they are, they're as distantly related as cacti are to blueberries, right? So even though they look very similar, and probably the first botanists that were finding them were thinking that they would maybe in the same family, they're actually very, very distantly related. So they have this convergent evolution. And when you think about independently evolved pitchers, we also wanted to understand if you have this convergent evolution of a plant host, to what extent do you see convergence of the associated communities? To what extent do we see that the same kind of things colonize these convergently evolved plants? So some of these, we knew that there were these different insects that tended to colonize, protozoa and rotifers, fungi and bacteria, mites, and generally in ants tend to be the same, the most common prey found in pitcher plants. It depends a bit on the different species, but generally ants are found as the most common prey. But we wanted to really dig into this and analyze it more in depth. So I'm just going to tell you quickly about what we found. Generally, we found that Nepenthes and Sarracenia pitchers are both colonized by organisms from the same phylogenetic clades. So what that means, this is a big evolutionary tree of all of these different bacteria. So it's showing the relationships, how they're all related to each other. And the brown on the outside it's showing kind of the relative abundances of all these different bacteria in our bog water or soil that were like directly surrounding where these pitchers were growing. So this was their kind of broader regional pool, right, of the different microbes that were around them. And then in red and in blue, you can see the Nepenthes and the Sarracenia, the organisms, the bacteria that were colonizing them. And to make sure that we were finding things that were, in good association with these pitchers, we only looked at things that were found in at least 10% of the different pitchers that we sampled globally. And so you can see these are the things that show up kind of regularly and repeatedly in pitcher plants. And they tend to come from related groups of bacteria, right? So these are different families of bacteria. And you can see that you have the red and the blue showing up in the same places on this big evolutionary tree. So the reason why it's important that we have kind of closely related things showing up in pitcher plants is that closely related things generally do more of the same functions, right? Like if you're in the same family of bacteria, you're more likely to be able to break down the same kind of compounds. You're more likely to produce the same kind of secondary metabolites, chemicals. So they're more likely to play similar ecological roles. And then for eukaryotes, it was a little more restricted, but we found kind of what you might expect, which is the dipteran insects, the histiomatid mites, and rotifers were found in both Sarracenia and Nepenthes in totally different parts of the world. So comparing North America and Southeast Asia, but you still find, even across the globe, that similar kinds of organisms are colonizing these pitchers. We wanted to know a little bit more about function and if maybe they have similar functions, right, happening in these pitchers, even on opposite sides of the world. So we particularly wanted to look for genes that were for prey degradation, right? We hypothesized that pitcher plants might have more chitinase genes, which chitinase is this enzyme that can break down chitin, which is that main biopolymer, the main part of an insect exoskeleton, right? It's not a very easy thing to break down. Not everything has chitinase genes. So we wanted to see if, compared with other types of habitats, if pitcher plant microcosms had more of these chitinase genes. And what we found when we compared pitcher plants, the Nepenthes and Sarracenia, with phylosphere, lake, and soil environments, that yes, on average, we saw higher amounts of chitinase genes in pitcher plants. On the other hand, we thought we would find lower cellulase genes because cellulose is a plant material, right? And probably the pitcher plants don't really want their microbes breaking down their plant tissues. And also, since insects are the main prey, we don't expect that that would be really enriching for things that break down plant tissues. And we did find that on average, also, pitcher plants had fewer cellulases than were found in these other types of environments. But what about Cephalotus follicularis? So at the very end of my PhD, I was able to go and sample from these little pitcher plants that are found only in Western Australia. It's also called the Albany pitcher plant because it's only found in this region near Albany in Western Australia. And I got in touch with a great botanist, Steve Hopper, who works at the University of Western Australia, and he was able to show me where a couple populations of these Cephalotus follicularis are, and they're so cute. They just have these beautiful colors and it's really neat shape, and it felt really special to be able to actually sample from these in the wild. I think it's not a lot of people have seen these pitchers in the wild. They're able to be cultivated, but it's really rare to see these natural populations. And some of them are so tiny. So on average, most of these are maybe about like 1 to 2 inches tall, 2 inches maybe on average, but you get these ones that are maybe like half an inch. And so those ones you can't really sample from. They're just too small. There's not holding a lot of fluid in there. But on average, these Cephalotus are probably smaller on average and hold a little less fluid on average than Nepenthes and Sarracenia, although, of course, it depends on the species. So we found that Cephalotus follicularis also host similar communities. When you look at the numbers of species of bacteria and eukaryotes in pitchers around the world, you compare them with Nepenthes and Sarracenia. On average, Cephalotus was like right around the other pitcher plants. We compared it with Nepenthes mirabilis that we had also collected in Australia because we wanted to have kind of an Australian connection, and then also the other Nepenthes and Sarracenia that I had collected in other parts of the world. And we compared these with things like, you know, water just collected in tubes set out, leaf water collected in leaves, bog and soil water. And in general, we found that pitcher plants tend to have fewer species of both eukaryotes and bacteria than in these other habitats. We also see that they tend to come from similar groups. So now this is similar to those phylogenetic trees I was showing you before, but this now includes the Cephalotus in green. And you can see again that they tend to show up in the same places as those red and blue. So you see the same kinds of bacteria also colonizing Cephalotus all the way over in Western Australia. And for eukaryotes, it's also more restricted, but we see the dipteran insects And we can also pick up the ant prey that are generally found in pitcher plants worldwide. So now we know that on this broad scale, pitcher plants generally host similar kinds of species. But when we compare these taxa more closely, do we also see host specificity? And what I mean by host specificity is like, does every different species of pitcher plant tend to select for a particular type of community that's specific just to that host, right? Do we see that if we look more deeply, not such broad categories like bacterial families, but if we look more closely, do we see that there are certain taxa that tend to colonize different species? And so to be able to look at this, you need to look at things that are growing together in the same habitat, but that maybe are all, from the same genus, like across different Sarracenia species. Do we see different host signals in the communities that colonize them? Do we see that in Nepenthes? And so I looked across these 8 different Nepenthes species and these 6 different Sarracenia species. And you can see that they look really different, right? They've got a lot of differences in terms of their coloration, their shape, their size. And we did actually find that within each genus, the host species identity influences the pitcher community. So it matters which host species these different microbes get into, right? It matters for what the community looks like. But what might be driving this? What are different characteristics of these hosts that could be affecting this? What we found was that different characteristics of these pitchers drive composition differences. So in the Nepenthes, we know that they have real strong differences in their pH or acidity from super acidic at 1.5 all the way up to around neutral. And that was what was really driving differences in their bacterial communities. So this is an NMDS plot. All you need to know about these plots is that each point represents like the whole bacterial community in one pitcher. And if they're close together, then those communities are more similar. But if they're far apart, those communities are more different. And they're colored by their pH. So over here is 1.5, and then over here is 7.5. And you can see that the pH structures the composition of these communities. For the Sarracenia, they don't have the same drastic differences in their pH, but they do have differences in their shape, where some Sarracenia are more kind of squat and round and hold more fluid, others are more narrow and tall. And we saw that that really mattered for the communities that live in them as well. And both of these things mattered more than the effects of dispersal or where they were in the world. So, if you're in Massachusetts or Florida, but you have the same shape, you're more likely to have a similar community. If you're in Borneo and Singapore, but you have a very low pH, you're more likely to have a similar community. We also wanted to dive more deeply into just looking at these Gulf Coast Sarracenia. So this is looking at this little area from like Alabama through to the Florida Panhandle. And we found that when we look at these, this was together with a PhD student in my lab, Jacob Pyle, we found that both host species and site matter. So it matters which host you're in, and it also matters where you are. For both your bacteria, the 16S is bacteria, or the 18S is the eukaryotes. And you can see the little stars show what's significant. So both host species and site were significant for both the bacteria and the eukaryotes. And then for the bacteria, we also saw that pH mattered, volume of the pitcher mattered, latitude and longitude all mattered. But for the eukaryotes, that wasn't as big of a factor. Those things didn't matter so much. When we looked a closer look at Nepenthes in a controlled setting, this was with Dr. Kadeem Gilbert, and maybe you've heard his talk. He gave a talk, I think, a little while ago. But this was at Hort Park in Singapore, where they grow many different species of Nepenthes together in the same glasshouse conditions. They filled the pitchers with the same pH 6.5 water, and then they sampled 2 weeks after the water was added. And what we found was that the pitchers are really able to change the pH of their fluids. They can excrete these hydrogen ions that acidify their pitchers. And so we got, from all the way from like 1 to 6 pH. And that really mattered for what communities, even after just 2 weeks, they're filled with the same water. And after 2 weeks, they can have drastically different acidities, and they have different communities that are structured partly because of that acidity. So the main takeaways is that pitcher plant communities across the world share many of the same species in their pitchers. The different species living in these pitchers likely play similar ecological roles in these little ecosystems. But when you look more closely, you also can see these clear differences in communities based on host characteristics and on geography. So which species matters and also where they are. In this second part, I want to talk more about understanding microbial functioning in the pitcher plant microcosms. And so I touched on that a little bit with the cellulase and the chitinases early on, but we want to dig into this a little more. And the first thing I want to talk about is potential nitrogen fixation by bacteria in Nepenthes. So some bacteria can actually take nitrogen out of the air, right? Air is mostly nitrogen. They can take it out of the air and actually make it into a usable form of nitrogen, like ammonia or something like that, that organisms can actually use in their metabolism. So we looked at 3 different species in Singapore and 4 different species in Borneo. And we saw this really cool pattern where generally, if pitchers were more acidic, they had less of this NIFH gene, which is a gene related to nitrogen fixation in bacteria. And so generally, if they were more acidic, they didn't have a lot of that NIFH, but if they were less acidic, they tended to have more of that NIFH, so more potential to be able to fix nitrogen of the bacteria. And we saw similar patterns also with the ones in Borneo. These were with our shotgun metagenomes, so we didn't have very many samples, but we saw the same trend where the more acidic pitchers had less of this NIFH genes present. And we think there might be this cool trade-off where pitcher plants that are more acidic actually use more of their own proteases, which is these enzymes that break down proteins to get the nitrogen in their pitchers. But if they're less acidic, they're a little more habitable for bacteria that can do this nitrogen fixation. And so there might be this cool trade-off where if they're less acidic, they're actually getting some more of their nitrogen from their bacteria that are living in the fluid, taking it out of the air. We don't know for sure because this is not showing actual function, but this is showing potential function. But it's a cool avenue I'd like to look into more maybe in the future. Working with a PhD student who's now a postdoc in my lab, Dr. Jessica Bernardin, we looked at if the way that bacterial communities function affects the pitcher plant host health. Like, does it matter for the plant which bacterial community it has? And so she did this controlled growth chamber experiment, inoculating pitchers with different bacterial communities that we knew had different functional profiles, different things they were able to do. We also used experimental controls, just deionized water, or also this acidified cricket media, which is also what we used to grow the bacteria on. And so she did many different assays of different physiology, including chitinase and protease that I've been talking about. We also did 16S metabarcoding to look at who was there, the composition of the communities. And then we did metagenomics and metatranscriptomics, which are other ways of also looking at the functioning. And at the end of this 8-week experiment, we measured plant traits, so the different, the size of the pitchers, as well as leaf nutrients and things like that. And this was just published 1 month ago. So if you want to read more about it, you can. What we found was that it really does matter for the pitcher plants which bacterial communities they have. So one bacterial community here, COM-B, on average, it grew pitchers 1.5 times larger. And that was just over 8 weeks. So it's a pretty short span of time, but it really affected the growth of these plants compared to the controls. So what might that be? We think that maybe it's that community happened to have more chitinase activity, was better at breaking down that chitin in the insect prey. And we had 2 different ways of measuring that, with just activity in a lab assay of actual chitinase activity, but also looking at differential gene expression with our metatranscriptomics. And here we have some different chitinases, and you can see that COM-B, which is the middle column here, tended to have more of these chitinases than the other communities. So we think that higher chitinase activity may be responsible in part for this increased plant growth. One thing I want to note is that the composition, like who was there in terms of the bacteria, was not really that different. So you can see here, this is showing different genera of bacteria present. You can see that a lot of the same bacteria are present across these different communities. And we didn't see that there was a whole lot of, specific taxa, specific species that were present in COM-B relative to the other communities. So it seems to be more this synergistic effect of the whole community rather than really just the presence or absence of particular species. And I think that's neat because it's like you need this whole community to be able to really have this important increased functioning. Jessica was also interested in if pitcher plant microbiomes function differently depending on what prey they catch. And so she ran this experiment controlling what prey. You can see the video she took here of a flesh fly being caught in a pitcher. So there, the pitcher is trapping that prey. And she carried out this experiment. It wasn't actually in a native range of the pitcher plants of Sarracenia purpurea, but it was in this high altitude bog in Switzerland where she was doing another study and was able to do this study at the same time. And the plants actually are very happy in Switzerland. So you get these big, big clusters of really large plants. And so she was able to use this block design to have introduced ants, beetles, flies, or no prey to pitcher plants, and then to see what happened. She also looked at how much protein was in the different prey types. So you can see here that flies on average had the highest protein, then the beetles, and then the ants. And we saw this cool relationship where the pitchers that had flies as their prey had higher protease activity on average over time. So we really did see that like the functioning of these communities, these microbial communities, was responding to what type of prey was added to the pitchers. But again, we didn't see very big differences in the composition of our communities. So there's a lot of overlap here in the composition of which bacteria were there in the different prey treatments. So what that shows us is that the community composition sometimes does not provide information about these important functional differences. And you actually have to measure the function and not just who's there. So the main takeaways for this part is that pitcher plant bacterial communities may fix nitrogen, right? Especially in less acidic pitchers. And this has been found to some extent in both Sarracenia and Nepenthes, but it hasn't been studied really in depth. And so we don't yet have a great sense of like how prevalent this is and how important it is for the nitrogen budget of the plant. But pitcher plant bacterial communities also clearly help the plants grow in other ways, right? So in this experiment, we saw that the pitchers grew a lot larger depending on what bacterial community they had. And it's probably related to prey digestion and nutrient acquisition in the pitchers. We also saw that it's likely to be the synergistic effects of the community rather than just the presence or absence of particular species. And that the composition of the microbiome does not always tell you about important functions. You need to find other ways to be able to measure those important functions. It's not really enough just to know who's there. So I want to end by just emphasizing that these tiny microbes have these really outsized effects. So they can be so important for the biogeochemical cycles of our planet, for our carbon, our nitrogen cycles, our, even the way that rain is formed and produced in different regions of the world. And every single leaf on every, large, even rainforest tree is going to be filled with different microbes. And those microbes have the ability to affect their large hosts in either positive or negative ways. And even when we think about our bodies, right, we have our skin microbiomes, we have our gut microbiomes, we are also hosting these microbial communities that have important functions for us. And I think studying pitcher plants, even though they might be this kind of unusual organism, they can help us to understand like how these communities form, what's happening there, how they function, and to get a better sense of the importance of microbes and how they relate to their hosts, even across different systems. I want to acknowledge a lot of different people that supported this research over the years, as well as places that allowed me to do field work in different parts of the world. And the funding that supported this, a lot of it was through the National Science Foundation, but also the James McDonnell Foundation, the Museum of Comparative Zoology at Harvard, the Simons Foundation, and thank you to many collaborators and students and trainees that have helped over the years. So with that, I want to thank you all for listening. and say that you can feel free to email me if you have other questions in the future. I'm happy to respond to emails. Sometimes I might be slow getting back to people, but I enjoy being able to talk to people about these kind of things. And I'm happy to answer any questions.
Thank you, Leonora. That was an excellent presentation. The people who attended this live thoroughly enjoyed it, and they have lots of questions for you. I did want to mention that our monthly webinars are free, but the International Carnivorous Plant Society does have a paid membership option. You can learn more about that by going to icps.clubexpress.com. A lot of universities buy memberships for their libraries. A lot of researchers are members, but we also have backyard growers and nursery people. When you become a member, you get access to our international seed bank. Our seed bank is only as good as what our members make it. So if you have surplus seeds, we would love for you to donate them to us. And I think the biggest benefit of being an ICPS member is that you get a quarterly newsletter called the CPN, the Carnivorous Plant Newsletter. And this has been around for 52 years. So not only do you get a quarterly newsletter, you get access to the last 50 years of newsletters. So you get to learn when a species was named, discovered, and not just species, but also cultivars. And you get growing tips, you get field trip reports, and then you also get updates on exciting events. So here's just some more photos of our full colored newsletter. Some of the exciting events are our Carnivores in the Classroom grant. This happens every August. This is also going to go into its 5th year this August 2025. It is to fund 50 teachers around the world with $150 worth of carnivorous plants for their classrooms. So if you know a kindergarten teacher all the way up to 12th grade who wants to add carnivorous plants to their classroom, please encourage them to apply. They have to make a 5-slide PowerPoint on how they would incorporate this. Like they have to create a lesson. It's very simple. So it could be about evolution. It could be about how to grow a plant if it's for younger students. It could be about plant propagation. Kids love to grow plants by seeds and cuttings and pullings. And there's lots of great content that you can teach using carnivorous plants. This year, 2024, these are the nurseries that donated those live plants around the world. We have nurseries, a lot of them are in the US, but we also have nurseries in Europe and Australia. And we're always looking for more carnivorous plant nurseries to donate to more classrooms. And that middle organization is Carnivorous, which is a company in Canada. And they designed 5 carnivorous plant for us. They are 3D models. If you have a 3D printer, you can print these for free yourself, or you can purchase them on their website. But if you go to our website, carnivorousplants.org, you can find a friend who has a 3D printer and print them yourself. And in that Carnivorous Plant newsletter, we also do really big announcements like our international conference. Every other year, we have a conference. We're going on, I think, the 14th or the 15th conference. We don't have a conference in 2025, but we will be in the Americas in 2026. We could be in North America, Central America, or South America, and we should be making that announcement coming up soon, maybe the spring of 2025. So you have 1 year to plan your international conference. All right, Leonora, we're going to post this on YouTube in another day or two, but I wanted to read the questions out loud so people know what we're talking about. One of the comments is from Stephen. He is very excited because now he knows about the communities within the pitcher plants, and now this is just, quote, one more thing to get to grow your plants better. And then Leonora, in the Gulf Coast, some of the Sarracenia species look very similar to one another. Other than the pH and the volume quality, are there limiting factors that influence the commensals that live within the same area, but what humans have decided are different species? Did you notice a difference between those
Yeah,
commensals?
yeah. So even in pitcher plants that are sort of the more like tall fluted ones, like flava or alata or rubra or leucophila, a lot of those we were sampling from too. And so they tended to look more similar to each other than to something like rosea or purpurea that has the little, cup and has
Wow.
more like easy to see liquid, right? It looks more watery. But even in those tall ones, we did see some signatures of host species differences in their microbiomes. And so that could be related to other things like, there may be some differences in the chemical profiles or in the like secretions that they excrete, or there may be some slight differences in the prey that they attract, or, there may be some other slight differences among them that also allow for some host specificity. And there is also been some cool research that has shown that when you kind of have pitcher plants that like grow in one region, but then maybe they expand across a river or into another state or something, that sometimes those organisms move with the pitchers, right? So you may actually see this like evolution happening over time together. So they found that with some of their mites, like the mites seem to sort of kind of co-evolve a bit with their hosts. And I think then some of the insects, right? Some of the dipteran insects, the flies and mosquitoes and midges. And then I think that might be true to some extent with some of the microbes, but it's hard to really know because they are environmental microbes. So they're also like in the surrounding environment and can kind of colonize at different times. So yeah, the different host species do matter. If they have more similar characteristics, they're probably going to also have more similar communities, but there may still be other aspects that kind of keep a few different distinct communities that may be co-evolving.
Jackie would like to know, did you sample from multiple pitchers on a single plant? And did you notice a difference or how much variation within one single plant did
Yeah,
you see?
that's a great question. Yes, and we do see strong differences within a single plant. And we think that the main reason that is because of succession over time. So on a single plant, the pitchers are produced at different times, right? They're not producing two pitchers that are exactly the same age usually, right? You have a range of ages. And so you have older pitchers and newer pitchers on the same plant. And even though they're so close together, they do look different. And some of the current research we're doing is looking at succession and how they change over time as the pitcher ages. And that actually is a really strong factor in what the community looks like, is how old that pitcher is. So I think that matters more than if they're growing on the exact same plant. How old the pitcher is really matters. And so you do see these distinct differences. We're also trying to tie it now to some functional differences because we want to know, like, how does the microbial community differ in how it functions over time as well?
Okay, I think this was when you were talking about your researcher in Switzerland. BJ
Mhm.
asks, when feeding the pitchers, were they given an equal number of insects or equal weight or equal amount of protein?
Yeah, so we standardize it based on mass, like on weight, basically, like how much um, biomass there was, right? But there were differences in protein because we know that the different kinds of prey had differences in their protein levels. So um, the flies had more protein than the ants, right? And we did see that reflected in how the microbial communities responded. Like we saw more of that protease activity happening with the flies when there was more protein present. So the communities do seem to be responding to what prey they're breaking down, and they're changing their function depending on what's there.
All right, I don't know when Lewis asked this, but speaking of Sarracenia in Switzerland, they say microbial communities are very interesting, but can they be considered, or can carnivorous plants be considered as a threat to the ecosystem if they're an invasive species? And I don't know
Yeah,
if
yeah.
don't know
So
if he
it's
knows this, but we're not
yeah,
very happy with the Sarracenia in Switzerland.
right, exactly. So in the areas that we were sampling them in, they kind of are treating them as naturalized at this point. They're like, there's a lot of them, and we're probably not getting rid of them, and the tourists really like them, so we're going to keep them. for the most part. But yeah, they want to make sure that it's controlled, and that they're not making other native species go extinct. And I think the places where I've heard it being the biggest problem is actually in the UK and some of their bogs, because they have these like heath bogs that have a lot of unique native species that are pretty sensitive. And it seems like in those regions, the pitcher plants might be having more of a negative impact on those ecosystems. So in a lot of the UK areas where they have the invasive pitcher plants, they are trying to eradicate them and going in and pulling them out. So yeah, it's very different than from what we see in the US.
All right. You have lots of, I really enjoyed this presentation. And then a lot of people were asking about your paper, which different uh, participants have already shared on your behalf. So thank you for that.
I can share some other links too. Yeah, here's one that I had in here. This was one of the early ones comparing the context-dependent dynamics. And I can try and maybe after this, I'll share a few more if people hang around. I'll try and put more links into there. I saw there was a couple that came just directly to me.
Okay,
So
perfect.
the first one was Mark Johnson. In addition to roles in digestion, the human gut microbiome plays significant roles in immune system function. Is there any likelihood of pitcher gut microbiomes influencing pitcher growth or behavior? I think there is, but we haven't dug into it that much, but I think there is potential for that. And in other like leaf-associated or root-associated microbiomes of other plants, they do see that the colonization of the plant tissues by microbes affects the plant immune system. And in a lot of ways, that might be one of the ways that they help plants not get attacked by pathogens. So if like a commensal or a mutualistic, like a beneficial microbe colonizes plant tissues first, it can prime the plant immune system to be ready to attack bacteria or fungi, even though it's not necessarily attacking those beneficial ones. But then if another pathogen attacks, the immune system is ready to fight it off. So that might be one way. They probably also help just by, filling up niche space and colonizing and making sure that pathogens don't have as much kind of like open niche space or nutrients to come in and take. And I think they could affect growth and behavior, but we don't know yet. But it would be interesting to know if a pitcher plant, maybe if it has certain microbes, then the volatiles it produces are different. I found that in a study of other plants that when endophytes were present in the leaves, the volatile profile was different. So that could also be happening, and that could change how it's attracting prey. There could be things like that that are changed depending on what microbes are there. Mark also asked, did I look at 18S tags of ciliates and or amoeba and look microscopically for protozoa? Yes, so that 18S did pick up ciliates and amoeba, and we saw some of those different ones that were present in different pictures all over the world. And then we've looked a little bit microscopically for protozoa in collaboration with like Tom Miller in Florida. He looks a lot at some of the protozoa that colonize. And actually I have a student who's on here today, Fiona, who's starting to look more at the protozoa as well. And we have some in culture. So yeah, the protozoa are they're often acting as predators of the bacteria. So they play this important role where they kind of keep the bacteria from going too much out of control. And then they're probably prey for the mosquitoes and midges. And so there's this nice food web that happens in those pitchers.
To kind of go off of that, Lewis asks, what defense mechanism do carnivorous plants have to prevent infections by these microorganisms? But most of your work was what we call commensals or,
Yeah.
mutualism.
Yeah. So I think in the pitcher plant case, these pitchers usually are pretty waxy and have like pretty tough cell walls on the inside of the pitcher. But they do have these digestive glands. And if you look like under a microscope, you can sometimes see that the bacteria or fungal yeast might cluster around these digestive glands a little bit. But we don't really see that the things that are colonizing the pitcher and living in that aquatic ecosystem are usually pathogens that affect the plant. So it doesn't seem like there's a lot of transitioning between commensal in the pitcher to like pathogen in the plant tissues, although that potentially could happen. But probably what's being controlled is, partly that like waxy, tough leaf surface is not super permeable for microbes. And then the digestive glands, maybe microbes could enter through those, but they're probably under more kind of plant control. And so I haven't looked to see if microbes can move through those digestive glands, but it's possible.
So I know that non-carnivorous plants have chitinase to fight off fungal attacks, but carnivorous plants use it to digest their arthropods and other things that are made out of chitinase. Do you think they also use that enzyme to fight off fungus, or is it
Yeah,
repurposed?
probably. I think what's happening is that for the Nepenthes, probably in their leaf tissues, they still have chitinases that they're using to fight off fungal pathogens. And then the types of chitinases that have been basically like co-opted into new types of chitinases now are the ones that are probably being excreted into the pitcher fluid and used to break down the insect material. So I bet the plants are doing both. Yeah.
Odysseus says, if you cut off the pitchers from a Sarracenia, how does the microbe community like recolonize? Is it the same as what you had seen? Are they just in the environment?
Yeah, my guess would be that you would get, as those new pitchers grow and mature, you probably would get pretty similar microbes colonizing them again. But yeah, if you're growing them in the UK or this, somewhere else in the world, they probably are going to have slightly different communities that are colonizing because we do see both an effect of location as well as the effect of the host species. And so I think where they are does matter for that regional species pool of what can colonize. But you probably will generally see similar types of organisms colonizing regardless where they are. So just the fact that they're this particular type of habitat that's aquatic and that digests insects, you're probably going to see particular types of organisms that like want to live in that type of habitat colonizing regardless of where they are. But to some extent, biogeography matters too.
Okay, Leonora, if we could do two more questions. Michael asks, how diverse are the rotifer species within Nepenthes?
Yeah, that's a good question. In the Sarracenia, we generally see these bdelloid rotifers. And those we did also see those in the Nepenthes. So they're in that same kind of group of bdelloid rotifers. But I don't have a really good sense of like how diverse they are. So I think they kind of fall into a particular group of rotifers, but other than that, I don't have a great sense. And yeah, rotifers are very cool, and it would be fun to know more about the species and if there's coevolution happening. I think in some pitcher plants there may be kind of those rotifers that just they move to new pitchers, then they move to new pitchers, they move to new pitchers, and so you do kind of get the same types of organisms living for a long time in the same type of pitcher habitat. But yeah, I don't know that much about them beyond that.
All right, for the last question of the day, Alberto asks, do you think it's possible to use Nepenthes as a biological identifier to understand environmental changes in specific location? Like, are they a good flagship species or
Yeah, that's a good
keystone
question.
species?
Yeah, yeah. Maybe, there may be bacteria that or fungi or protozoa that particularly like to live in pitcher plants. And so the way that I think about it mostly though is that with nitrogen deposition, with people draining bogs or making parking lots on top of pitcher plant habitats, we're losing these pitcher plants. And when there's nitrogen deposition, they stop making those round pitchers, right? They just make these phyllodia that don't have that pitcher environment anymore. So I think when that happens, when the pitcher plants aren't there, you may really be losing associated species as well. It's not like you're just losing the plant, you're also losing those organisms that need this habitat to survive. So I guess in a way, I think they're kind of a keystone species because they're creating a habitat. And so if they're gone, their whole habitat is gone and everything that relies on that habitat is gone. I think it's maybe less likely that, they do have some, like the pitcher plant mosquito, is only found in pitcher plant habitats, right? And that might be true for microbes as well. But that's less known. Like we don't really know so much about if there are particular microbes that are only found in or only thrive and complete their full life cycle in the pitcher plant habitat. But I do think, yeah, that they are keystone species in the sense that they are creating this whole habitat for a lot of other organisms.
All right, very good. Thank you, everyone who attended this webinar live, and we look forward to seeing you all in 2025. And thank you so much, Leonora, for a great talk. And we hope everyone has a great day. This will be posted on YouTube in a day or two with all the links that Leonora is about to send me.
Okay, I will stay on for a bit and I'll put in some other links. Thank you all for listening. It's really fun to have great questions and to have a good audience. Appreciate it.
It's not a surprise that gardeners, educators, and scientists are fascinated by these unique plants. The International Carnivorous Plant Society, or ICPS, not only love these plants, but welcomes growers just getting started all the way through professional scientists. The ICPS even started an annual World Carnivorous Plant Day to celebrate them. The free online event is held the first Wednesday of May. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.